Nobody else got it.
Except for he's like the whole, oh my god. Go figure. Welcome to another Biostatistics lecture series. The topic today is the multiple comparisons. If you haven't already, please sign up in the back, and there is also an evaluation sheet. If you look on the back side of the evaluation sheet, there's a disclosure, so that they have to have that on there. Also, on the first slide and the last slide of the handouts, there's actually our next upcoming lectures, so just make note of those. And if you didn't get a handout or need an evaluation sheet, just raise your hand and I will come around and get that to you. And today's speaker is Brent Logan.
Thank you, Kelly. So I'm going to be talking about multiple comparisons, and before I get into that, I just want to disclose that I don't have any relationships. So I'm going to start off with an introduction and a motivating example. We'll kind of review hypothesis testing so that you can understand where multiple testing or multiple hypotheses comes into play. We'll talk a little bit about the philosophical issues. Should you be correcting for multiple tests? Should you not? And under which circumstances? And then we'll go through a series of specific examples which will help you see the breadth of the potential issues in multiple comparisons. So multiple testing, the subtitle of this talk is Torturing the Data Until It Confesses. And that's the basic idea. So if you get nothing else out of this talk than that, then you hopefully will have a pretty good handle on the problem. If you look at the data long enough, you look at enough outcomes, look at enough comparisons, you're going to find something. It's going to tell you something. And that's the basic fundamental problem here. So let's talk about a motivating example. This is a panic disorder study to measure effectiveness of a drug treatment compared to a control group. And the investigators were interested in studying 4 outcomes. So they, those 4 outcomes were the severity of anticipatory anxiety, total number of panic attacks, severity of phobic avoidance, and the global assessment of the patient. And you can see the 4 p-values from those comparisons. The first p-value is 0.04 for the severity of anticipatory anxiety, and the other ones are 0.1, 0.72, and 0.38. So if we take our typical way of doing hypothesis testing, then the conclusion that we would make from this study, based on deciding that there's a difference between the treatment and the control if the p-value is less than 0.05, is that the drug has a statistically significant effect on the severity of anticipatory anxiety. So what is the multiplicity problem, or how does that come into play here? Well, basically, the problem is the 4 outcomes were studied. We looked at 4 different outcome variables. And once you look at multiple outcome variables, once you do more than one test, you increase your chance of finding at least one false significant result. So the question that this raises, the fact that we looked at 4 outcomes, is whether the impact of treatment on severity of anticipatory anxiety is real, or whether it's simply an artifact of performing 4 tests. And you can consider, an extreme setting where you just keep examining more and more outcome variables until you find one with a significant p-value. And that's where the torturing your data until it confesses comes in. So to understand the framework for how multiple testing is done, I want to review a little bit about hypothesis testing first. So with hypothesis testing, we usually specify a null hypothesis and an alternative hypothesis. The null hypothesis often refers to a hypothesis of no difference between 2 treatments, or no difference between groups, or maybe a hypothesis of no association between your treatment and your outcome variable. The alternative hypothesis is typically your research hypothesis. It may be that there's a difference or an association between groups. So the way that hypothesis testing is often done, you compute a p-value, and that p-value refers to the likelihood of obtaining an observed difference, or one more extreme than that, if the null hypothesis were true, or the likelihood of obtaining that difference by chance alone. And then you make a decision based on that p-value. You decide that you would reject the null hypothesis if the p-value is less than a significance level of alpha. So that's how standard hypothesis testing works. And you can think about the types of mistakes that you make in terms of this kind of a table. And you may have seen this kind of a table before. There's a truth about the population. Okay, the null hypothesis could be either true or it could be false. And then there's some kind of decision that you make about that hypothesis. You might accept the null hypothesis or reject the null hypothesis. And in fact, if you accept the null hypothesis and it's true, then that's a good decision. If you reject the null hypothesis and it's false, then that also is a good decision. If you reject the null hypothesis and it's true, that's a bad decision. And if you accept, and that in fact is called a type 1 error. So that's the significance level of the type 1 error that you would have seen in reading the literature. And the other bad decision that can be made is a type 2 error, where you accept the null hypothesis, but in fact it's not true. So your significance level is the chance that you make a type 1 error, okay, that you reject the null hypothesis or you find a difference when there's really none there. And the type 2 error rate is beta, and it's the complement of your power. So what happens when we do multiple hypotheses? Well, suppose that we do a certain number of independent tests with a type 1 error of 5%. Well, if we do each one with a type 1 error of 5%, well, the impact of the multiple testing can be seen if we look at one of two things. If we can look at the chance that we make at least one type 1 error among all the tests, all the k tests, or the expected number of type 1 errors that we make among all of those k tests. Those are two ways that we can think about what's the impact of testing multiple hypotheses. And so you can see here in this table, if we just do one test, then the chance that we make at least one type 1 error is just 5%, and our expected number of type 1 errors is just 0.05, so about 5 out of 100. But if we do two tests, the chance that we make at least one type 1 error goes up to 10%. And if we do 10 tests, the chance of making at least one type 1 error, at least one false finding among those 10 tests goes up to 40%. And you can see as you get up to 100 or 1,000, the chance of making at least one type 1 error when each of those tests is done at a 5% level goes up to basically 100% chance. So you're almost certain to make a type 1 error if you don't make any adjustment and you do lots of tests. Now if you look at the expected number of type 1 errors, you can see here, for example, if you do 100 tests, you'd expect about 5 of those 100 tests to be type 1 errors if they're all true hypotheses. If you go up to 1,000, you'd expect 50 type 1 errors among those 1,000 tests, and so forth. So you can see that when you start looking at multiple hypotheses or multiple tests, there's some other considerations that you may want to keep into account in terms of your error rate or your expected number of errors. So we'll talk about this a little bit more formally shortly. So what are the implications for our example? Well, we did 4 tests. So if those were all independent tests, we would have about an 18% chance of making a type 1 error using that 5% cutoff. Okay, that's an 18% chance of obtaining a statistically significant result, even if there was no effect of the treatment on those outcomes. So with that context, that p-value of 0.04 may seem less convincing given this context of doing 4 tests. So the question here is, how do we actually formally incorporate the impact of multiple testing on our inference? So there's a lot of strategies, and some of them are conceptual in nature, some of them are statistical in nature. The first one, very simple, do less tests. If you do less tests, your tests are more convincing, you don't need to, the need for adjustment is much less. Another important consideration is transparency. Transparency in the number of tests that you do, so that other people can evaluate, reviewers of a journal article can evaluate what you're doing. And people may have different strategies for dealing with the multiple testing, and the transparency helps other people to consider that. Being cautious in your interpretation when you're doing lots of tests. That's certainly an important consideration. One ad hoc kind of adjustment that people sometimes use is just to use a significance level of 1% rather than 5% when you're doing a whole bunch of tests. Again, this is very ad hoc. It doesn't really control a particular kind of error rate, but it reduces the chance that you make a false significant finding. So in terms of the formal statistical criteria for dealing with multiple hypothesis testing, there's two basic strategies. The basic strategy is to control a different error rate. Okay? So the error rate that we were controlling when we were only doing a single hypothesis test is the chance of making a type 1 error on that one test, okay? Of rejecting the null hypothesis when it's true. When you're doing lots of tests, there are two criteria that are fairly standard in the literature in terms of accounting for multiple testing. One is the family-wise error rate, which is the chance that you make at least one type 1 error among all the tests that are performed. So it's basically penalizing you for any mistake, any type 1 error that you make across all of the tests. And the other one is called the false discovery rate. So the false discovery rate is the expected proportion of false discoveries out of all of your significant findings. Out of all of your rejected null hypotheses, what fraction of them are false rejected null hypotheses? So it allows some type 1 errors, in a sense, as long as they're not a large fraction, as long as it's a small fraction of all of your rejected hypotheses. And you can see this in terms of these, this kind of a table. Now this kind of a table looks very similar to what we had before, except what we're putting inside these, the entries of the table are not yes or no, it's a good decision, but the number of hypotheses that are falling in that decision. Okay, so for example, A would be the number of hypotheses that you tested that are true, but you accepted them. So that's, again, a good decision. Okay, B is the number of false null hypotheses out of all that you're testing that you accepted. So that would be a bad decision, and so forth. So you get the same kind of idea here. So A and D are the number of hypotheses in those two good decision situations, and B and C are your two bad decisions. The number of hypotheses that you're making bad decisions about. So with this kind of a table, what are those two error rates that we talked about here? The family-wise error rate, that's the chance that you make any type 1 error, that you take any hypothesis, any true hypothesis, and reject it. So that means C is 1 or more. Okay, you made any kind of a bad decision in that section there. Okay, any of the true null hypotheses got rejected. Okay, so that's what the family-wise error rate refers to. The false discovery rate is kind of like an expected value or an average of the ratio of C over N. So C is the true null hypotheses that are rejected, and N is the total number of rejected hypotheses. So as long as the fraction of your rejected hypotheses that are true, as long as that fraction is small, then you're controlling your false discovery rate pretty well. Okay. So how do we control the family-wise error rate? Well, there's again, a number of strategies that you can do that. One common strategy is to conduct an overall test. For example, and what that overall test is going to be different depending on the situation that you're in. And I'll go through some examples. But basically an overall test of whether there is an effective treatment on all the outcomes at once. Okay? And so you would only examine those individual outcomes if that overall test was significant in the first place. Okay? If it's not significant, then you don't bother to look at the individual comparisons. Okay? So it's kind of like a gatekeeper strategy. You only proceed past the gate and look at each of the other outcomes if you find an overall significant difference. Now that particular strategy gives you what's called weak control of the family-wise error, which is control only when there's no difference on any of the comparisons. Another strategy, another common strategy, is to basically adjust the significance level, or equivalently your p-value, for the number of tests that you're performing. So for example, if we are testing k different hypotheses, then a Bonferroni procedure basically says use a significance level of alpha divided by k, or 5% divided by the k hypotheses being tested. Equivalently, you can multiply all the p-values by k and then compare those directly to your significance level, your unadjusted significance level. Now, in terms of adjusting the significance level for the number of tests that you're performing, there are other more efficient procedures available. You probably want to talk to a statistician about those. But this strategy actually gives you strong control. It controls the family-wise error rate, the chance of making any type 1 error in any situation. Okay? Maybe one of the outcome variables is very different between treatment and control, and none of the others are different. Well, it still controls your family-wise error rate for that kind of a situation. In terms of controlling the false discovery rate, so the Benjamini-Hochberg procedure is a pretty standard procedure for doing this kind of control. So a 5% false discovery rate, again, on average, 5% of your significant findings will be false significant findings, or true hypotheses, a true null hypotheses. So what you do to apply this procedure is first you order the p-values, and then you compare the ith smallest p-value to a threshold of I times 0.05 divided by k. So we'll look at an example of this. Here's an example comparing two groups on a psychological scale, which actually has 14 different subscales. Okay, so we're going to do a comparison of the groups for each of those 14 subscales, which entails doing 14 tests. Okay, so if we split our 5% type 1 error rate into 14 pieces, then we get our Bonferroni significance level of 0.0036, which is on the far right column. Okay, so here I give the ordered p-values, and you can see what that threshold is. And so you can see here that you would reject, under a Bonferroni procedure, you would reject the first 4 hypotheses because they're all below that Bonferroni threshold. But you wouldn't reject the remainder of the subscales. If you apply the Benjamini-Hochberg procedure, you can see that the threshold starts at the Bonferroni threshold, but then as you go up to larger p-values, it actually increases up towards 0.05. And so what that gives you is it gives you an easier threshold to get past as you start to reject more and more hypotheses. In fact, so what you look for is what p-value is still below. You look for the largest p-value that's below that threshold, and you reject all the hypotheses with smaller p-values than that. So here you can see that you're rejecting the first 7 hypotheses with the smallest p-values. So you can see applying a Benjamini-Hochberg procedure is more efficient, more powerful than a Bonferroni procedure because you're going to reject more hypotheses. On the other hand, you're controlling a different error rate. Okay, the Benjamini-Hochberg procedure is going to allow some type 1 errors into the hypotheses that you're rejecting, as long as they're a fairly small fraction. Whereas the Bonferroni procedure won't allow any type 1 errors. Okay, so that's a basic illustration of how those two procedures work. In terms of should you be using a family-wise error versus a false discovery rate procedure. A false discovery rate controlling procedure is generally more powerful than a family-wise error controlling procedure. It's going to be more likely to detect real differences or false hypotheses, false null hypotheses. Again, it allows for an acceptable rate of type 1 errors among your significant findings, but it's not appropriate, the false discovery rate is not appropriate when you want strict control over any type 1 errors across all of your tests. So the false discovery rate is naturally suited towards an exploratory setting, and is also very appropriate in a large-scale multiple testing situation, like when you're doing genetic microarray analysis or an imaging, where you're looking at a lot of points in an image. Family-wise error controlling procedures, again, strict control of the risk of any type 1 errors across all of your tests. And it's more appropriate, especially in a confirmatory or in a regulatory setting, such as a phase 3 clinical trial, or something that's going to be approved by the FDA. So we've talked about hypothesis testing. What about the confidence intervals? And the little cartoon says, when you say you're 95% confident, just what are you inferring? So it's important to always, consider the confidence intervals. Okay, it's not enough to simply give a p-value and say it's significant or it's not. Whether or not you adjust for multiple testing, the confidence intervals gives you an idea of the magnitude of the effect. And so, how do we deal with confidence intervals when we're doing lots of tests or when we're estimating lots of parameters? Well, we can think about the equivalence between hypothesis testing and confidence intervals here. One way of testing a null hypothesis, say a null hypothesis that a mean difference is 0, would be to construct a confidence interval for the mean difference and see if that confidence interval contains the null value of 0. Okay, so if the confidence interval is entirely above 0 or entirely below 0, then that would tell you that 0 is not a plausible value for that mean difference, and so you would reject that null hypothesis. So there's this equivalence between hypothesis testing and confidence intervals. And in fact, that equivalence bears through to the type 1 error rate versus the confidence level of your interval. In fact, the type 1 error rate of your hypothesis test corresponds directly to the complement of your confidence level of your interval. So 5% versus 95%. So how do we deal with confidence intervals for multiple parameters? So now we're talking about intervals for a difference in one outcome and a difference in another outcome and a difference in a third outcome. So what we can do is we can construct simultaneous confidence intervals. So what's the difference between a typical confidence interval and a simultaneous confidence interval? A 95% confidence interval tells us the probability that interval contains the true value is 95%. A 95% simultaneous confidence interval says that the probability that all the intervals contain the true parameters is 95%. So it gives you a much stronger statement about the coverage of those values. The simultaneous confidence intervals will be wider than the individual confidence intervals, but they have this direct correspondence to the family-wise error rate. Okay, so if you adjust your hypothesis test to deal with multiple comparisons, you really should adjust your confidence intervals to reflect that as well. Okay. So let's talk about a little bit of the philosophical issues around adjusting for multiple comparisons before we go into a series of examples. As you might imagine, there are differing opinions on whether or when adjustment for multiple comparisons is needed. There's a lot of considerations in terms of making that decision. One of the concerns about adjusting for multiple comparisons is that the choice of the family of hypotheses that you're adjusting for is arbitrary. Is it these 4 outcomes, or did we look at another 8 outcomes on top of that? Well, which one do you want to adjust for? And in fact, the results of that adjustment for multiple testing are very sensitive to what family of hypotheses you're adjusting for. So that's certainly one concern, that it's somewhat arbitrary how that adjustment gets done. But in general, a strategy of choosing small, more focused families of hypotheses, specified a priori in writing if possible, will help you get around that arbitrariness and avoid the potential for kind of cheating the multiplicity adjustment. And that's where the transparency comes in as well. Another concern about adjustment is there's an increase in type 2 errors when you do a multiple comparisons adjustment. So what this basically means is you have a loss of power or the likelihood to detect real differences between groups when you use a multiplicity adjustment. And you can see that. If you adjust the significance level from 5% down to 1%, you make it that much harder to reject the null hypothesis, and you bring your power down. Okay? So there are appropriate and powerful testing procedures, but oftentimes these are particular to certain situations. So it's important to see a statistician of that if control of the type 1 error rate is important, but you want to maximize your power or reduce the loss in power that comes from that adjustment. People that argue against adjustment often propose that you should do unadjusted analysis, but with full disclosure of the data analysis procedures. The counter-argument against that is that when you don't do the adjustment yourself, it becomes difficult for the reviewers to evaluate, actually. So there's some pros and cons to all of that. In particular, the need for adjustment and the best method of adjustment is often scenario dependent. Okay? There's a number of things that you want to consider when you're deciding whether to adjust for multiple comparisons and how. One thing is it plausible that many of the null hypotheses might be true? Okay? If you think a priori that many of those null hypotheses might be true, you want to account for that and make sure that you're not just getting significant findings because of all the hypotheses, all the potentially true, likely true hypotheses that you're testing. Another consideration, do you want to ensure reproducibility? Okay? If you do a lot of hypothesis tests, you don't adjust, and you get some significant findings, if you do the study again, those may disappear. Okay? So the best way to ensure reproducibility is to deal up front with that multiple testing issue. So if you want to be able to claim that an identified significant finding is in fact real and is reproducible, then you need to adjust for that multiple testing. Are you trying to heavily mine the data to find a significant result? If you're doing that, you certainly need to consider the multiple testing aspect. Is your study expensive and unlikely to be repeated before serious actions are taken? Then you need to consider the cost of making a decision with or without adjusting for that multiple testing, and consider that reproducibility of that result. And in particular, is there an important cost associated with type 1 errors? If there is an important cost, then you need to consider strict control over any type 1 errors rather than a false discovery rate type procedure, which allows for some type 1 errors. So let's look at a number of scenarios in which you might need to deal with multiple testing. One of them, as I've alluded to in my introductory example, is that of multiple outcome variables. So again, the first strategy to consider, limit your number of outcomes. Barring that, if you want to control the type 1 error, there's a couple things to consider. Those outcome variables are often correlated. So the Bonferroni method itself can be conservative in that situation. And so there are more powerful procedures available. You want to talk to a statistician about those. And overall, another strategy, so that we also talked about the strategy of doing an overall test. So an overall test of no difference on any of the outcomes is generally more powerful than treating each outcome separately. And those are done with a class of procedures called multivariate methods. So in our panic disorder example that we started with, if we do a Bonferroni adjustment, you can see the p-values and then the adjusted p-values, which basically is done by multiplying by 4, the number of hypotheses. And once you do that, then none of these outcomes are significant after adjustment. None of those adjusted p-values are below 5%. So none of them would be significant when you control the family-wise error at 5%. You also could, in this scenario, do an overall test. Okay, so an overall test here would mean that the null hypothesis is there's no difference between treatment and control on any of those 4 outcome variables that we are studying. Okay, and we're doing it with one test. Okay, and the p-value from that kind of a test, it's a multivariate test. That's actually called a Hotelling's t-squared test. The p-value from that is 0.2. So because that overall test is not significant, we would not really want to proceed to look at each of those individual outcomes. This kind of a gatekeeper of an overall test prevents you from getting into this multiple comparisons problem. So again, because of that, we would state that there's no evidence of a difference on any of the outcomes. Okay, so because that overall test is not significant, we would want to ignore the significant p-value on the severity of anticipatory anxiety. That's likely attributable to this issue of multiple testing. In particular, multiple outcome variables are sometimes dealt with in confirmatory clinical trials in which you want strict control of the type 1 error rate. Now, oftentimes in confirmatory clinical trials, the strategy is a little bit different here. At that point, in many cases, you can specify a primary endpoint that you want to focus on in that trial. There's usually one of them. It's pre-specified in the protocol. And so you bypass this issue of multiple comparisons by basing your inference on the primary endpoint in a confirmatory clinical trial. Now, there's often secondary endpoints in those clinical trials, but they're considered exploratory, or they may be explanatory of the findings in the primary endpoint. But they're not, you couldn't use those secondary endpoints except if you explicitly talk about it to justify approval by the FDA for a drug. So because you have usually one pre-specified primary endpoint, there's no multiplicity adjustment typically used. Sometimes there's not a clear single endpoint, but you can piece them together. Composite endpoints is another way of bypassing this multiple testing problem. You can combine multiple endpoints into one to reduce that multiple comparisons problem. So some examples of that: cardiovascular setting. The events that you might be interested in studying between the 2 drugs would be myocardial infarction. You might be interested in stroke, cardiovascular death. So you can combine those outcomes into one outcome and save yourself from the hassle of dealing with multiple comparisons. In the cancer setting, you might be interested in progression of the disease or in death. So you can combine those into progression-free survival. So it saves you on the multiple testing issue. It may cause problems in terms of interpretation of the results. For example, if the treatment improves one outcome and makes the other one worse, well, then you have a problem there, and it's difficult to interpret. Another kind of deviation from that issue of multiple outcomes is where you have multiple time points. So a repeated measure study, this is an example where you're looking at dental measurements in boys and girls at ages 8, 10, 12, and 14. So I've plotted the means, the mean response profiles for boys and girls. The boys are on the top of this line. And so you can see, you can see the profiles here. So you may be interested in, are boys and girls different? And if so, at which of these time points? Okay, so again, if we were to do separate t-tests at each of those 4 time points, comparing boys versus girls, we have this susceptibility to multiplicity issues. So there's a couple strategies to get around that. Sometimes with repeated measures data, you might fit some kind of a linear growth curve. So that reduces your number of comparisons from 4 different time points to maybe 2 parameters, like a slope and an intercept. So you're fitting a line to that data. So that reduces your multiple testing problem from 4 to 2, basically. You also could do a strategy of doing an overall test, where your null hypothesis says that there's no difference between boys and girls at any age. So then you only do comparisons at each of the individual ages if that overall test is significant. And then you could also do this Bonferroni adjusted p-value. So you compare the boys and girls at each of these stages. So you can see the p-values given here at age 8, 10, 12, and 14. P-values are 0.066, 0.084, 0.005, and less than 0.001. So with an unadjusted strategy, we would find a difference at age 12 and age 14, but not at age 8 and 10. If we do a Bonferroni adjustment to those p-values, basically multiplying by 4, we still find a difference at age 12 and age 14. So in fact, you get similar findings with an adjusted and unadjusted analysis. If you do the overall test, the null hypothesis is that there's no difference between boys and girls at any age. The p-value for that overall test is 0.006, and you can get that from a repeated measures ANOVA test. And since that overall test is significant, then you could look at those unadjusted p-values at each time point. But again, the findings are the same. You get significant differences at age 12 and at age 14. So all these multiplicity adjustments basically give similar findings. So your final conclusion here would be that the mean dental measurements are significantly different between boys and girls at ages 12 and 14. Another scenario where you would need to deal with multiple testing is when you're dealing with multiple groups, okay? More than 2 groups, for example. This is a study comparing B leukocyte counts in 51 subjects with colorectal cancer. So there's 4 classifications of cancer patients that were studied here. In terms of those B leukocyte counts, there's a Duke's classification system which gives you classes of A, B, or C. And then there's a 4th group, group D, which refers to patients with disease which had not been completely resected. So we have 4 groups, and there's actually 6 pairwise comparisons that you can do among those 4 groups. Okay, A with B, A with C, A with D, B and C, B and D, and C and D. So this is what the data looks like. These are box plots of your B leukocyte counts. So you're showing the medians and the interquartile ranges and the ranges. It's suggestive of potentially a difference between A and the other 3 groups, but it's certainly not conclusive based on the box plot. And you can construct those unadjusted p-values for these pairwise comparisons. So I've set that up in a table here. So A versus B, the p-value is 0.045. A versus C is 0.098. A versus D is 0.062. And then B versus C is 0.683, and so forth. So based on those unadjusted p-values, if you don't make any adjustment, then you would find a significant difference between classification A and classification B, because the p-value is 0.045. Now, how would you do an overall test? Or an overall test here says that there's no difference in B leukocyte counts between any of those 4 groups. Okay. So and you can do that with an overall test using analysis of variance. If you're doing non-parametric testing, you can use a Kruskal-Wallis test. But the analysis of variance F test has a p-value of 0.91. Okay, so even though we have that p-value of 0.045 for the comparison of A versus B, the overall test is telling us that there's no difference among any of those 4 groups. Okay, so this strategy would say that you should ignore the significant result in those pairwise comparisons when that overall test is not significant. So your final conclusion, no significant difference in the B leukocyte counts. Now, if you want strict control of the type 1 error rate in this multiple group setting, the standard method, and you may have heard of it, is called the Tukey test. The Tukey test is actually a little bit more powerful than a Bonferroni test for the multiple group setting, for all pairwise comparisons among groups. But that gets back to that strategy of you may want to talk to a statistician if you're using a more sophisticated technique, which is more powerful. The adjusted p-values using that Tukey test are given here, and you can see that none of them are below 0.05. So once you do that adjustment, there's no longer any significant differences among the groups. So we get basically the same finding whether we do an overall test first or whether we do a Tukey adjustment. Another situation in which multiple testing is a problem is in the area of subgroup analysis, in particular subgroup analysis in clinical trials. The basic question when you're doing subgroup analyses is whether the effective treatment in a clinical trial is homogeneous across all the patients in that trial, or whether there's particular subgroups of patients where the effect of treatment is different than everybody else. This is kind of a classic example of the pitfalls of subgroup analysis. It's the ISIS-2 trial it was published back in 1988. There were 17,000 patients with acute myocardial infarction randomized to a placebo versus aspirin. There was also a streptokinase arm, but we'll ignore that for purposes of this discussion. The mortality within 1 month was 9% with the aspirin group and 12% in the placebo group, with the p-value of less than 0.001. You can see when you have 17,000 patients, you can find very small differences to be significant. But the investigators were urged by the editors of the journal that they were submitting to conduct nearly 40 subgroup analyses. The investigators actually agreed on condition that they could conduct their own subgroup analysis to illustrate the unreliability of the subgroup findings. So here's the subgroup analysis that they added. Astrological sign, okay? Libra, and so when they looked at these 12 different astrological signs, basically the month that you're born, they found in the Libra or Gemini group, there were actually more deaths in the aspirin arm than in the placebo arm. Whereas in the other groups, you had a significant improvement from aspirin. So the basic issue here is once you start looking at subgroups, you get increased variability of the results just by chance. Okay? It's not because astrological sign explains the effect of aspirin. That's highly unlikely. But you get increased variability results just because you're looking at 12 different groups. Furthermore, you're looking at 12 smaller groups, so that you get more variability as a result of that. Okay? But this situation is actually an excess of type 2 errors because of the multiple comparisons. Type 2 errors means you're not finding differences that should be there. Okay? It's unlikely that there's not an effective aspirin in the Libra or Gemini subgroups. So this would be this non-significant finding would be a type 2 error. So the multiple comparisons can impact both situations in a subgroup analysis context. Here's another example of subgroup analyses. This is the effect of a new versus a standard antibiotic on febrile morbidity in 4 age strata, as well as overall. So you've got age 20 to 24, age 25 to 29, 30 to 34, and 35 to 39. So if you look at the rate ratio of febrile morbidity, 1.4, 1.2, 0.3, and 1.1, with an overall rate ratio of 0.9. Okay? Here, you look at your confidence intervals. The confidence intervals encompass 1 for age 20 to 24, for age 25 to 29. It's below 1 for age 30 to 34, and it encompasses 1 again for age 35 to 39. The overall rate ratio, the confidence interval encompasses 1. So that would be suggestive of no difference between the new and standard antibiotics overall, and no difference in 3 of those age groups. But there's an indication that maybe there's a difference in the age 30 to 34. Okay, so again, if we were to analyze these 4 subgroups, we have this inflation of our type 1 error rate. There's a chance that the confidence interval is below 1 because we're looking at 4 different groups. We have more variability across 4 different groups here. In fact, so the best way to assess those subgroups is to perform an overall test of whether the subgroups differ. Okay? So here, what that overall test means when we're talking about subgroup analysis is that the treatment effect is the same for each subgroup against an alternative that the treatment effect is different for 1 or more subgroups. Okay? And this test is basically an interaction test. The null hypothesis is that there's no interaction between the subgroup and the treatment. So we would only look at each individual subgroup if that interaction test was significant. So here the interaction test had a p-value of 0.103. There's no significant evidence of interaction here. So the subgroup finding in the age 30 to 34 group is likely due to chance and the fact that you're doing 4 tests. Subgroup analyses in general are discouraged and because they're prone to overinterpretation of significant or non-significant findings. General recommendations about subgroup analyses: perform the interaction test, okay, and only look at each subgroup separately if that interaction test is significant. You want to confine your subgroup analyses to primary outcomes. You want to limit your number of subgroups. If you start doing subgroup analyses and you start doing it on multiple secondary outcomes, then you're just drastically increasing your multiple testing issue. The subgroup analysis really should be pre-specified in the protocol as much as possible. They lend that much more strength if they're pre-specified to the finding. You want to consider biological plausibility. Certainly the astrological sign doesn't have any biological plausibility, but it's just an illustration. And you should report all the subgroup analyses that were done so that there's transparency of how many you looked at. And that means you may need to adjust for multiple subgroup variables. Maybe you're looking at subgroups by age, by sex, by disease status. Okay, there's another layer of multiplicity when you look at multiple subgroup variables. And they should generally be considered exploratory. Another area is the issue of large multiplicity problems. So what is a large multiplicity problem? Well, one example is genetic microarray data analysis. Here you have thousands, maybe even tens of thousands of genes that are being assessed for their association with an outcome, a phenotype. There's a lot of ways of analyzing this kind of data, but some of them involve hypothesis testing for each gene individually. And when you do that, if you do each test at the 5% significance level, you would expect 50 false findings in 1,000 tests. So you can see that there's clearly going to be a lot of false significant findings here. So how do you deal with that is an important issue here. Another example of this large-scale multiplicity problem is in imaging, in particular functional magnetic resonance imaging, which is the example that I'm going to show here. Here you would perform an experiment and you would assess a signal. In the fMRI context, it's a blood oxygen level dependent signal, which indicates whether there's activity in a particular point in the brain. And you assess that signal at each of thousands of voxels, or even tens or hundreds of thousands of voxels, or points in that brain. So I'm going to be showing an example of a finger tapping experiment where you go in the scanner, you tap your finger, you stop, tap your finger again, stop, and so forth. It's a very basic experiment, not particularly interesting from a clinical standpoint, but it gives you a nice illustration of the multiple testing issue. So the null hypothesis here is that the mean bold signal while you're tapping is the same as the mean bold signal when you're not tapping. Okay, so one way to assess that again is a t-test. You can do a t-test at each point in the brain. And when you do that, you can put those together in terms of an image. Now, I've included not just the brain, but also the surrounding image outside the brain. You could certainly screen that out. But you can see, you can kind of see the highlight of the brain here as well. And you'll see it a little bit better when I show the next slide. So this is your t-statistic image. So they're scaled so that you get either dark red or dark blue in the middle when you have t-statistics close to 0. So findings that are not likely to be significant. And they're scaled towards yellow if they're positive, very large positive values, or light blue if they're very large negative values. Very, large and absolute difference, negative values. So what we're interested in is the two extremes, the light yellow and the light blue, in terms of finding an effect of finger tapping on a particular point in the brain. So if we threshold it at a 1.96, which is your usual critical value for a significance test with a 5% level that's not adjusted for multiple testing, this is the image that you get. And you can see that I've superimposed the image of the brain on here. And you can see, certainly there are a lot of false positives. There's false positive outside the brain, which is certainly one indication that they're not likely to be real effects. But there's just a lot of extra points that are above that threshold. So that's the issue of false positives here, of multiple testing. If you do, this is a 64 by 64 image, so there's 4,096 tests. And when you threshold at a 5% level, you're going to expect 5% of those to be, even if there's nothing going on in the brain, you'd expect 5% of them to be falsely rejected. If we do a Bonferroni adjustment here, you can see it gets rid of essentially, or almost essentially, all of those false positive signals. You get rid of all that noise in the thresholded image. And you can seem to pick up a bit of a signal here on the two, and those are going to be the cerebral motor cortex areas, which is associated with movement. But it's not a very wide area. So you're catching some signal there, but that's a pretty strong, the cerebral motor cortex is a pretty strong effect. If you do a false discovery rate threshold using a Benjamini-Hochberg procedure, you can see that you pick up a larger section of those areas. You get a couple of false signals in that image, but it's fairly small, fairly scattered, and you're picking up more of the signal. So that should help illustrate a little bit of the trade-offs. If you don't do any adjustment, you get a lot of noise. If you do a Bonferroni adjustment, you get rid of all the noise, but you also get rid of maybe too much of your signal. And if you do a false discovery threshold, you allow a little bit of noise as long as it's not much of what you're finding. Okay. And you get better power to pick up more of the signal. So that's the main benefit of doing this false discovery rate procedure. Unadjusted method, too many false positives, family-wise error adjustment, loss of power, and the false discovery rate procedure is basically a compromise in this setting. So the last setting that I want to talk about is this issue of interim analyses. So clinical trials are routinely monitored for safety by a data safety monitoring board. They meet every 6 to 12 months to review the data. And there's a lot of justifications that they might have for early termination of the study. High toxicity, problems with accrual, but there also may be a plan in the protocol for stopping for efficacy. If you find what appears to be a good result early, you don't want to have to continue the study to the end. You want to be able to report that, and ethically, it's important for patients. Futility is another consideration as well. Well, what happens if you do repeated tests for efficacy? If you look at your data halfway through the study or a couple times throughout the study and test it without making any adjustment, well, you're going to inflate your type 1 error rate. Okay? So here's an example. If you just look at your data once and test it with a 5% significance test, then your type 1 error rate is really 5%, as it should be. So if you just look at your data at the end of the study, then you have no problems. If you look at your data twice, maybe halfway through the study and then again at the end, the chance that you make a type 1 error is now 8%, okay, because you didn't make any adjustment for the fact that you're looking multiple times. You're doing multiple tests. 3 times, if you look at the data 3 times, it goes up to 11%. If you look at the data 10 times throughout your clinical trial, it gets up to as high as 19%. Okay? So multiple looks at your data will increase your type 1 error rate. Okay? So that's an important thing to consider. So when you're planning, so when you want to do this kind of multiple looks at your data, you really need to specify that plan in the protocol. And there are ways of adjusting for multiple looks at your data. Basically, it involves adjusting the significance level that you use at each interim analysis as well as at the end of the study. Here's an example of one way you might adjust that significance level, and this is based on an O'Brien-Fleming boundary, which is a fairly standard one that's used in the clinical trials. So at the first interim analysis, maybe after a third of the patients are evaluable, you would use a significance level of 0.0005. So there's a chance of stopping, but it's very small if there's no effect. So you require a very small p-value to stop at that point. Second interim analysis, you would use a p-value of 0.014. And then at the final analysis, you would use a p-value of, you would use a threshold of 0.045 for the p-value. So you can see that you're paying a penalty. If you're allowed to stop at one of those two looks, you no longer can use a 0.05 threshold at the end of the study. You need to be a little bit more stringent to account for that potential inflation of the error rate. So there's a number of ways of constructing these adjustments to the significance level. The O'Brien-Fleming is one popular one. So I want to just close with a couple comments in summary. The basic gist of the multiple testing issue is that the more tests you do, the more likely you are to find a significant result and increase your type 1 error rate as a result. So there's a lot of ways you can deal with that, but you want to be aware of the problem. You want to restrict or prioritize the number of tests that you do, first off. Be cautious and aware when interpreting results when you do a lot of tests. And then, if it's suitable, explicit corrections for multiple testing are available. They strengthen the evidence and the reproducibility of significant research findings. There is a potential loss of power, which could be a concern in an exploratory context. But when you do those corrections, you need to specify an appropriate family of hypotheses, an appropriate type of error rate in order to set that up. So that's all that I have. I just want to remind you of the resources that we have. The Clinical and Translational Science Institute supports education, collaboration, and research in clinical and translational science. There's a website. And the Biostatistics Consulting Service has comprehensive statistical support, and there's our website for that. There is free drop-in consulting, and here's the schedule, and this should be in your slides as well. And the upcoming lectures are also at the end of the talk as well. We put them in twice. So, thank you. Be happy to take any questions if anybody has anything.